Let's move on to the other one, Vaccinia striformis. A fleck to a 4, and some in between. And I think it's quite clear that all of these would be considered low. The 3 is not as susceptible as the 4, but I would also call that susceptible in my system. You can disagree, or depending on how you're classifying.
Is that yellow rust or straight rust?
Both.
But the color is not very yellow.
It depends on the settings of the camera. And it is actually yellow rust, Vaccinia striformis. And some of the reactions at the bottom. I use 0 to 4. And for my purposes, I think that's sufficient.
Yeah, so we normally use this 0 to 9 scale. And the advantage of that, we think, is that we have intermediate as well. And sometimes we can differentiate some reactions based on intermediate as well.
Yeah, 0 to 9 is very common. So you can decide in your situation which one will suit you best. This picture comes from the McIntosh et al. Book, the Gene Atlas. And I think quite clear that those 5 would be low and that one would be high. How would you rate this high intermediate on the right there? This one. There's some background chlorosis. You can see in between the pustules. So there is some response.
So the second leaf from the right, you're calling it low.
I'm asking the group.
It's actually compatibility. It's indicating compatibility. There is some resistance gene which is compatible with this.
Okay.
I would probably call it a 3 double plus or something, or maybe even add a C if there's a lot of very clear chlorotic tissue. Robert, how would you score that one?
The score is low, but again, you're using a seedling phenotype ultimately to predict the field phenotype. So it's very important to try and correlate that.
Yeah.
Ultimately, if it's in a breeding context with what's going on in the field.
But if this is in a differential set, and somebody in Australia calls it low, and somebody in Pakistan calls it high,
Yep.
you reach a different name.
Yeah, pathotype, absolutely. And that happens. And sometimes people with many, many years' experience argue about whether things are low or high.
Sure.
It's not often that clear cut.
So the best solution would be to repeat and repeat and repeat until you're pretty sure that you've got the distinction.
Consider the whole experiment, so you have the control as well.
But Zach told one thing, that is that you have got a check, so you can compare it.
Yeah.
Because when you go for physiology of the host, then there is something like source and sink mechanism. Because the pathogen, it draws nutrients from the adjoining points, and that results in that type of symptom. But practically, it is susceptible.
The other, can I just make one final point on that, Zach? So we know, for example, on race determination, we know, for example, a gene like YR1 is an infection type like the leaf on the left, right? If you take an isolate from the field and you put it onto a differential set and you get a response like the second one from the right on the differential for YR1, your conclusion would be that you have virulence for YR1. Possibly there's something in the background just giving a little bit of incompatibility, but the conclusion with respect to virulence on the gene is it's clearly virulent, because typically YR1 would give it a mutant.
Yes, you must also know the phenotype of the individual genes.
And you need to know the phenotype.
And then we have also other differentials with the same genes. So like we have Evosed 17 and VPM.
Sure.
And we compare about, so like VPM give this one, we term it 6 on our scale. And then this, if we look at Evosed 17, we have clear infection, so we term it as virulent. And this is the case of warrior race. So it goes on VPM is 6, 7, but on Evosed it goes compatible.
All right, good. So you can see there is some subjectivity in it, but I think as long as you have what they call controlled subjectivity and you're consistent in what you're rating, and you have your checks and you can explain what you're recording. Just a few examples. Again, you may say this is leaf rust, but it's not. It's actually Buccinea striformis. It's just the setting of the camera. Clearly low, am I right? And some genotypes give a very necrotic, very strong hypersensitive response, although you get a fair amount of sporulation. That's susceptible, that's susceptible. Low. And those two, I think, are pretty clear. One more, I think it's the same system. Those three are low, high, high, and those two low as well. Just to give you an idea in the stripe rust system how these infection types look. This is stem rust. High or low? All of them are low except this one. What would those two pustules be up there? That not stem rust? It's probably leaf rust contamination. Or a different race, some contamination. But that would be the typical high response. A more practical example from real greenhouse situation, it's quite easy to then, I think, distinguish your low infection type, your fleck, with a full susceptibility when you're doing segregating populations, for instance. This is Agelop sharonensis, so it's not wheat, but it's infected with Buccinea graminis forma specialis tritici. And you get the same range of infection types, although they look slightly different. What would you call, I think the others are clear, that's susceptible. These are the two parents, so this is a segregating population, by the way. But what would you call that one? Do I hear low? I think it's low. Pustules are fairly big, but you see a very strong sclerotic island surrounding those larger pustules. So we can use the same system for different hosts infected with rust, or the wheat rust anyway. Another wheat stem rust example, this is the SR9H gene. That's how it is expressed. It could be a 1 or a 2 on the 0 to 4 scale. And compared to your susceptible checks, quite clearly you can see the difference. Low versus high. These are pairs with different races. The names of the genotypes I don't think are important. But again, you can see low versus high. What would you call that? I mean, it's low, clearly. If you would like, you'd probably give it an X, and probably an X minus or X double minus, because the majority of the pustules are smaller. That one? It's low. It's probably a fleck 1C, even an N if you want. There's a lot of chlorotic and necrotic tissue. Then you have an intermediate size one without the typical islands, but clearly smaller than your susceptible. And it's not the textbook 2, but it is probably in the 2 range somewhere. The textbook 2 has what? It's got a green island surrounding the stem rust pustule. Just some more examples. We don't have to go through all of these. Low, 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 and again, a typical X. Some interesting ones there. What would you call that? It's low. It's also in the intermediate range. Flag 1 plus maybe, and a 4. This is an interesting slide. Wheat stem rust on wheat rye substitution lines. So these are not very typical wheat stem rust infection types. That one would probably be not too out of line, but this one, and that's not a mixture of isolates. You get these full susceptible pustules on the same leaf with fleck. So it's X. It's a very interesting response. That was the first time in my career that I actually saw this.